This animation is on what happens to a protein between translation and transport to the Golgi. As you are aware, this is a ribosome, consisting of one large and one smaller subunit. The ribosome is the main component of translation, and is the site at which protein synthesis occurs. The messenger RNA has a codon which is complementary to the anticodon of tRNA. This triplet code includes an amino acid, which joins the tRNA molecule to allow elongation of the polypeptide chain.
Once the peptide bond has been formed between two amino acids, the tRNA detaches from the mRNA in order to allow the next tRNA to bind. In this example, as soon as the protein has been fully translated, it migrates from the ribosome towards the rough ER. However, proteins may be translocated in the ER during their synthesis on a membrane-bound ribosome. Once in proximity to the rough ER, the protein then enters via a pore in the RER membrane. The construction of the polypeptide chains into the correct 3D conformation occurs within the rough ER. This is assisted by molecular chaperones. One of the major family of molecular chaperones are the HSP70 family. These are thought to bind to the unfolded polypeptide chain as it crosses the membrane, and then mediates folding of the protein and assembly of the multi-subunit proteins within the ER.
Inside the ER, proteins are glycosylated on specific asparagine residues. Oligosaccharide units of 14 sugar residues are added to the acceptor asparagine residue on the growing polypeptide chain. This process is catalyzed by oligosaccharide transfer membrane-bound enzyme. Another modification that occurs within the ER is the formation of disulfide bonds, which is catalyzed by disulfide isomerase. These disulfide bonds play important roles in the assembly and secretion of cell surface proteins, adding structural integrity and support to the protein.
Proteins that are correctly formed and have completed their specific modification processes are packaged into vesicles and then exit from the ER via ER exit sites, which appear to be randomly dispersed throughout the rough ER. These sites can be identified through a light microscope due to the lack of ribosomes on the ER membrane. Vesicles that have left the ER are enclosed in a COP2 coat, which consists of two subunits, shown here in yellow and red. Some of the membrane proteins trapped by the coat function as cargo receptors, binding soluble proteins in the lumen.
Once vesicles have left the ER, they shed their coat and begin to fuse with one another. The fusion of membranes in the same compartment is known as homotypic fusion. Homotypic fusion requires snares. The interaction is symmetrical with V snares and T snares, contributed by both membranes. The snare proteins are pulled apart by the NSF protein. The separated matching snares on adjacent membranes interact, leading to membrane fusion and the formation of one continuous compartment. The structures formed from the two vesicles are known as vesicular tubular clusters. These clusters are relatively short-lived, as once formed they begin to bud off themselves. Unlike the COP2 coated vesicles from the ER, these vesicles are COP1 coated. However, the structure of these two coats are quite similar. These vesicles from the vesicular tubular clusters travel quickly along microtubules to the Golgi apparatus, where they fuse with the cis face of the Golgi, delivering their contents. Proteins that enter the Golgi undergo further modification to suit their purpose. They then bud off from the trans face of the Golgi and travel to their intra or extracellular target.
Thank you for watching.
Created using Powtoon.